Fantastic. Okay, everyone, I apologize. I was trying to make sure that this was getting recorded. So I do want to go ahead and get started. I am Eva Buckner, and I am an assistant professor and the medical entomology state extension specialist at the University of Florida. So the reason why I'm interested in resistance testing and giving this webinar today is because I've been conducting mosquito research for about 10 years now. And 3 of those years was at a mosquito control district where I helped conduct research to be able to make operational decisions. And so that's really where I got introduced to insecticide resistance testing using the CDC bottle bioassay. So since moving on to my position at UF, I get to work with mosquito control programs throughout Florida and then elsewhere, actually international programs as well, and I'm able to teach them how to perform this assay as well. So I'm really excited about that, and I'm excited about this opportunity. Additionally, my lab currently has a grant from the Florida Department of Health to perform testing of Aedes aegypti, Aedes albopictus, and Culex quinkofasciatus populations from the state of Florida to determine their resistance status to multiple active ingredients. So this is certainly something I spend a lot of time about. So welcome, and we'll go ahead and get started. So just want to get started with some housekeeping. This is actually out of date. There was, when I last looked, there was about 150 people registered for this from the US and outside the US. And so within the US, we've got folks from Florida and many other states. So I welcome you all. I'm very happy to have you all attending. I hope you find this to be beneficial. Because we do have so many people attending, and unfortunately, I'd like to think that we all have the best intentions. It's impossible to know that. And just to make sure that hopefully there's no one gonna try and Zoom bomb us. You're not gonna be able to use your camera or microphone. So the way you're gonna be able to communicate is using that chat function. So just to make it easier to answer the questions, please just limit the using the chat just to questions if you don't mind. So my webinar co-host, Julie McConnell, who is Bay County Extension faculty, she's going to be monitoring the participants. And if someone is misbehaving, she will be able to kick that person out. Of course, I don't expect that to happen. And I'm not the most technologically savvy person. Let me just go ahead and get that out. So just in case I have technological difficulties or you have technological difficulties, this is being recorded and it will at a later date be available on the extension tab of FMEL's webpage. May do some editing, so give me about a week to get that up there. And I'm also going to post all of the resources discussed in this webinar on that tab as well. So certainly I cannot give this talk without recognizing Janet McAllister, the co-creator of the bottle bioassay protocol. And many of the slides you're going to see in this presentation were originally created by Janet, or they're going to contain images copied from the CONUS manual by both Janet and Cassie Scott. I'm not sure if Janet was able to join us on this webinar, but I know I did see that Roxanne Connolly from the CDC was able to join us. And so I have asked Roxanne and anyone else from the CDC to please chime in if you hear me say anything that needs to be corrected, or if there's anything you'd like to add. You know, I certainly just thank you. Feel free to do that, and certainly feel free to answer any questions as they come along. All right, moving forward. Just to give me an idea of how this webinar is going to run, first we're going to get that sign-in survey out of the way. I'm going to give some background information on resistance and the bottle bioassay, the bottle bioassay kit, and of course, so very important, how to go about getting it for free. Then I'm going to talk about the specifics of the bottle bioassay and show you a demonstration video. And lastly, go over how to interpret the results of the bottle bioassay and give you an example of how I have used the results to make adulticide suggestions to a mosquito control program. So here is the link to the sign-in survey, and there's also a QR code. Oh, I'm not sure. There's also a QR code. So if you have a smartphone, you should be able to just use the camera function on your smartphone and just, it will register the QR code automatically, and it should take you right to the survey. This would be beneficial if there's multiple people watching the webinar from the same computer, especially for those people that are attending this webinar and they are wanting to get the 2 CEUs for the public health pest control license in the state of Florida. You certainly have to fill in the sign-in survey as well as the exit survey that'll be provided later on. If there's any issues or questions about that, how to access the survey, please let me know in the chat, and I will be looking for potential issues in the chat right now. Okay. So I'm just going to give everybody just a couple of minutes to fill out the sign-in survey, and then I will move forward. Oh boy. Text. Okay, so I'm seeing that quite a few people are unable to access it. The sign-in survey, I really don't know why. So I will try and, yeah, I'm about to paste the link in the chat. So if you'll give me one moment to do that. So I just posted the link to the sign-in survey in the chat. So hopefully you'll actually be able to access it now. I certainly apologize for you not being able to access it via the PowerPoint presentation. So please let me know if you're continuing to have issues. I already warned you, I'm not so savvy at technology, so I don't know why it didn't work through the PowerPoint presentation. I apologize for that. Okay, so hopefully everyone has now been able to access the sign-in survey. Oh, hey, thanks, Carrie, making me feel better. Thank you. So hopefully everyone has now been able to access it, and going to, awesome. Thank you, Winona. Okay, all right. So moving forward, let me go here. I think I'm gonna need to re-show the PowerPoint presentation. Okay, so hopefully now you can see the next slide. I'm sharing my screen. I'm gonna try and make sure I can access this questions. All right, this is a learning experience for me, but we're gonna get going now. Thank you for your patience. So let's start at the beginning and talk about what insecticide resistance is. It's going to be a genetic change in response to selection by toxicants that may impair control in the field. It's believed to develop largely because of natural selection of pre-adaptive variants that possess genetically controlled mechanisms or other means of survival in the presence of an insecticide. This is just some examples of what can cause insecticide resistance, and specifically on this slide, I'm talking about resistance due to mosquito control activities. But if you look here on this little diagram, you can see that resistance in mosquitoes is certainly not going to be just to the actions of vector control or mosquito management or even urban pest control. It's also going to be due to the actions of homeowners and businesses as well as agriculture, because they are also going to be using pesticides and other chemicals that can potentially lead to resistance in mosquitoes as well. But just talking about the insecticide resistance, that that can be caused due to mosquito control activities. These include using a single class of insecticides, using a product that has long residual action, using a product with slow release formulation, or applying products to all life stages and all generations, and then also treating all habitats where the pest occurs. So just basically completely bombarding the mosquito and its different life stages and habitats can certainly lead to insecticide resistance increasing in that mosquito. So as I mentioned, insecticide resistance is certainly not going to be just to the due to mosquito control, and there are insecticides, herbicides, fungicides, and fertilizers that have similar structures as insecticides that are used in mosquito control. And so because of these similar structures and these chemicals, it can lead to resistance being created in the mosquitoes. And of course, you think about, I think about Culex quickofasciatus, or we call it the southern house mosquito here in the U.S. It likes to it likes to use storm drains as larval habitat. And so if you think about after a storm, there's lots of rain that's going across the landscape and picking up who knows what type of chemicals as it's moving across the landscape, and then that water and all those chemicals are going down into the storm drain, and then those mosquito larvae are being exposed to all those chemicals. So that's just one example of how resistance can be caused in mosquitoes that's unrelated to mosquito control. And certainly through the resistance testing project, my lab's been conducting testing populations throughout Florida. I will provide resistance testing results to mosquito control program managers, and the result will be resistant to a certain insecticide, and they'll say to me, but we've never used that insecticide. And so this is potentially why that is seen. So we talked about what can lead to resistance. So let's talk about potential mechanisms responsible for providing resistance in mosquitoes. And there's 4 of these: behavioral resistance, cuticular resistance, target site resistance, also known as KDR is really the biggest one that we talk about, metabolic resistance. So for behavioral resistance, this is going to be maybe a mosquito changes the time that it's host seeking, or trying to get blood meals from humans, so that it's not going to be interacting with insecticides when they're applied in the evening, for example. So with cuticular resistance, it could be thickening of the mosquito's outer cuticular layer that can slow insecticide penetration into the skin. And then, of course, with target site resistance, there could be mutations in the voltage-gated sodium channel protein to which the pyrethroids bind to, and reduce the chemical's ability to recognize their target. And this is actually illustrated here. And then metabolic resistance is illustrated here. And in this example, there's enhanced activity of the metabolic enzymes. And in this example, it's going to be cytochrome P450s, or CYPs. And the CYPs are one of 3 enzyme classes that are able to break down pyrethroids before they kill the insect. So while these are 4 mechanisms responsible for resistance seen in mosquitoes, it's the target site resistance and metabolic resistance are going to be the two primary mechanisms seen, and metabolic resistance is actually going to be seen more than target site resistance. So I mentioned that there are 3 classes of enzymes that are capable of providing metabolic resistance, and these are also known as detoxification defense enzymes. And the 3 classes are oxidases, esterases, and glutathione S transferases, or to make life a whole lot easier, GSTs. So while we talk about KDR and we talk about metabolic resistance as these being mechanisms that are potentially responsible for causing resistance, they're not happening completely independent of one another. And certainly you can have multiple mechanisms occurring in the same mosquito that's going to be contributing to the resistance that you see. And so that's what this illustration is showing here. Again, I'm going to have to thank Janet McAllister for this graphic. Thank you so much, Janet. Moving on, just to give you a couple of other terms for you to familiarize yourself with. Cross resistance results from a common detoxification system or from target site insensitivity. And then we also have multiple resistance, and this extends to a variety of classes of insecticides with differing woof. Okay. Just kidding. Okay, so I think my camera just messed up. So I'm gonna work on trying to get that fixed. So I'm gonna work on getting the video fixed, but I'm gonna continue on with the presentation. So insecticide resistance in a mosquito population is going to initially be detected and characterized by using some type of bioassay, such as the WHO adult bioassay that you can see pictured here. Additionally, you could use the topical bioassay. Field cage tests are another great option to discover insecticide resistance. And then of course we have the CDC bottle bioassay. So these are all potential tools for detecting mosquito insecticide resistance. Fabulous. So let's talk more about the CDC bottle bioassay. It's a surveillance tool, like I said, just one of the surveillance tools, but certainly a very beneficial one for detecting insecticide resistance in mosquito populations. And I know that I'm talking about mosquitoes because mosquitoes are pretty much my life. But of course, this bottle bioassay can be used to detect resistance in other insect populations, other vector populations, but I'm just gonna talk about mosquitoes, but just know that this tool can be used to detect resistance in other vector populations. So the CDC bottle bioassay is designed to detect an active to determine if an active ingredient in an insecticide formulation is going to be effective against a mosquito population at a specific location at a given time. So basically, you can think of this as being a snapshot of how effective an active ingredient in an insecticide formulation is going to be. Because it is a snapshot, you should conduct bottle bioassays on a regular basis to continue getting that surveillance information. It's generally recommended to conduct these CDC bottle bioassays for about once a year. So now that I've described what the CDC bottle bioassay is, I certainly think it's important for you to know about the CDC bottle bioassay kit that the CDC is so generously providing for people that are interested in conducting resistance testing. So all the supplies here that you see in this photograph are provided in the CDC bottle bioassay kit. And so you're going to get disposable plastic pipettes, mouth aspirator, tubes for measuring acetone and insecticide solutions, of course the protocol for conducting the bottle bioassay, the data sheet, and of course safety data sheets. But just to let you know, there are materials that you're going to need to provide yourself. So container for transferring and holding mosquitoes, timer, permanent markers, labels, and all safety equipment that you need. And then, of course, the mosquitoes that you're going to be using for testing. So how can you request the CDC bottle bioassay kit? So it's going to be different for folks in the US and outside the US. And I just took a screenshot of this web page here. You can see the web address here, so you can, if you want to go to this website later. But for international programs, you're going to email bottle bioassay at cdc.gov to order your bottle bioassay kit. And then for programs within the US, you're going to order your supplies by emailing US bottle assay kit at cdc.gov. So that's really exciting, finding out how to get free stuff, especially for conducting resistance testing. And just to let you know, on this webpage that I've taken the screenshot of, in addition to getting the bottle bioassay kit, you can also obtain for programs that are outside the US, you can also obtain protocols for conducting the bottle bioassay in 3 different languages, so that can certainly be very helpful. And then additionally, there's an instructional video that's available on this webpage. So for within the US, you can access the CONUS manual. Of course, everyone can access this, but this is a version of this bottle bioassay protocol that came out this year, and it's for evaluating insecticide resistance in mosquitoes using the CDC bottle bioassay kit. So it's all the steps within the protocol are written specifically for using the tools, or sorry, the everything that you're going to be given in the free bottle bioassay kit. So I think that's really helpful if you're just getting started and you don't know what to do. You can get your free supplies and you can follow everything word by word, step by step in this manual. So where you can, and this is the webpage where you can find the manual. As I mentioned at the beginning of this presentation, I'm going to make sure that all of the materials that I talk about are going to be available on FMEL's webpage. They may not be up there immediately, but hopefully by the end of the week, I'll have all this available for you. If you're not able to access this now off of the PowerPoint presentation, I'm going to make sure that you can obtain this later on. So now that we've covered what the bioassay is and where you can get your supplies for conducting your own resistance testing, let's go ahead and talk about how the bottle bioassay works. So the CDC bottle bioassay relies on time mortality data, which measures the time it takes an insecticide to penetrate a mosquito, make it through the tissues of the mosquito, and get through the target site and act on that target site. And here you can see an illustration of that process in a susceptible mosquito. So the black dots are going to be representing an insecticide. So the insecticide is making it through. It's certainly going to, even though this is in a susceptible mosquito, there's certainly going to be processes occurring in the mosquito that are going to decrease some of the pesticide or insecticide from making its way to the target site. But some of the, because it is a susceptible mosquito, some of the insecticides are certainly going to make it to the target site and act on that site. But this is not going to be an immediate process. This is something that takes time for to occur. So in comparison, we have a similar illustration for this process of an insecticide making its way through a resistant mosquito, and anything that prevents or delays an insecticide from achieving the objective of killing insects, so making its way to that target site and acting on that target site, it's going to be contributing to resistance. So you can see here we have similar, some of the same processes that you saw occurring with the susceptible mosquito. You're still going to have excretion, but it's also going to be more than what you saw with the susceptible mosquito. And there's going to be additional mechanisms that we talked about. So detoxification, target modification. And so these are going to be mechanisms that are causing resistance, and more time is going to be needed for an insecticide to reach the target site in a resistant mosquito. And so that additional time that is needed is represented by this yellow arrow here that's tagged on to the time that's needed for the insecticide to make it to the target site in a susceptible mosquito. So here is an actual graph of mortality for a susceptible mosquito, time to mortality for a susceptible mosquito population. And here is the illustration you saw on the previous slide. So now this is just showing an example of the potential time that it could take for this to occur in a susceptible mosquito population. And of course, this it can vary by mosquito species, population, insecticide, but this is going to be the time that is needed for 100% of mosquitoes, of susceptible mosquitoes, to die at a certain dose of an insecticide. And this is known as the diagnostic time. So that's one of the important terms with the bottle bioassay is being able to know what the diagnostic time is that you're going to be comparing your your field mosquito populations to. And when I mention the diagnostic dose, I'm going to talk more about that later, so don't stress about what that means. But I should say, actually, you can use different diagnostic doses, and really the diagnostic dose is going to be the concentration of the insecticide solution per bottle within the bottle bioassay. And in my lab, we just use the diagnostic doses suggested by the CDC, and you're going to see those later in a chart. So we talked about the time that's needed for an insecticide to make it to the target site and act on the target site, leading to death in a susceptible mosquito population. And we have an example time that this amount of time that this could take here in green. And as I said, in resistant mosquitoes, they're going to potentially need, actually they will need more time for that insecticide to act on the target site. So any mosquito population that's surviving beyond this diagnostic time is diagnostic time threshold is considered to have some type of resistance. So this diagnostic time here, again, the amount of time needed for 100% of a susceptible mosquito population to be killed by an insecticide. This is really the defining point between susceptible and resistant mosquitoes in that you can compare your field population or within your field population, because certainly, oh, let me go back. Because certainly within that, if you have a field population that, and it may or may not contain resistant mosquitoes, that's the whole point of conducting the bottle bioassay, but should it contain resistant mosquitoes, it's possible, you can also have mosquitoes within that population that are also susceptible. So that is what this graph is just trying to illustrate, that there's going to be susceptible and resistant mosquitoes both within the population, and using that diagnostic time is how we determine that the percentage of the population that is going to be resistant. So in this example, only 30% of the field population was susceptible, because they're the only ones that died by this diagnostic time. So the CDC bottle bioassay detects differences in mortality at the diagnostic time between susceptible and resistant mosquitoes. So that mortality rate at the diagnostic time for a mosquito population, it's going to determine whether or not, it's going to determine the resistance status of that mosquito population. And this chart is illustrates the different categories of the resistance status categories. So you perform the bottle bioassay, and then you can, at the diagnostic time, you're going to see what the mortality rate of your field population is. If you have more than 97% mortality at that diagnostic time, then that population is going to be considered susceptible. If it's between 90 to 96% mortality, then that's going to be considered developing resistance. And then anything below 90% mortality at that diagnostic time is going to be considered resistant. So those are the three resistant statuses to be aware of. But and this is an example going back to the graph you saw. You can see that using these different groups, the population, the field population that we've been looking at, because there's only 30% mortality at this diagnostic time, then certainly this field population is going to fall underneath the category of being resistant. And so certainly it's important to be able to look at the mortality rate of your field population at a diagnostic time to determine whether or not that population is resistant or not. But it's also important not to just stop the bottle bioassay at your diagnostic time. You want to continue running your bottle bioassay through 2 hours, because while the mortality rate at the diagnostic time is really important, you also need to see what the mortality rate is at the end of the 2-hour bottle bioassay to determine the strength of the resistance in the population. So if 100% of the mortality is achieved at some point beyond the resistance threshold or the diagnostic time, for example, here, that's going to be represented by the resistant number 2 population, the light blue line here. That resistance mechanism is not going to be as great or as strong as the resistance mechanism or mechanisms that are being seen in this resistant number 1 population illustrated here with the purple line. Because while you don't have 100% mortality at the diagnostic time for your resistant 2 population, you do achieve 100% mortality during the bottle bioassay, so at 90 minutes. But for this resistant number, sorry, I may have mixed up the numbers there, but for the purple line, the resistant number 1 population, 100% mortality is never achieved. So the highest mortality rate during the duration of the 2-hour bottle bioassay that is observed is 40%. So you can certainly see that the resistance mechanism is going to be higher in the resistant number 1 population compared to this resistant number 2 population. So that's why it's important not just to make note of the mortality at the diagnostic time, but also certainly the mortality at all the different time points beyond the diagnostic time and at the conclusion of the 2-hour bottle bioassay as well. So I think it's important to point out that when resistance is detected with a bottle bioassay, it doesn't necessarily mean that you're going to see control failure in the field. And that's because the bottle bioassay is really not designed to, that's not what it was designed to do. The bottle bioassay is designed to be super sensitive, and actually it's supposed to allow you to determine or to detect resistance developing in a population before you're going to lose the use of that chemical against the population. So I like to think of it as the bottle bioassay alerting you to resistance that's occurring in your population before that you're going to see that failure. So maybe like canary in the coal mine. And because the bottle bioassay is not designed to tell you if you're going to see control failures, you can see here the suggested algorithm provided by the CDC for further testing depending on the level of resistance detected by the bottle bioassay. So in our example that we were talking about, the mosquito population that exhibited, well, actually both of them exhibited mortality rates of, I think it was 40% or something. So they certainly fell in the resistant category. But you want to continue on and conduct other tests to determine the intensity of the resistance mechanism. And then of course, you can also conduct mechanism testing, and I'm going to talk more about that later. But to be able to actually know whether or not you're going to see failure in the field, it would be necessary to conduct field testing. And should you see that you're not going to be getting the level of control that you want using the field testing, then certainly it's a good idea to consider alternative control methods. So I just want to provide you with a couple of considerations before conducting bottle bioassays. So thinking about what diagnostic time and diagnostic dose to use. So we talked about diagnostic time. It's the amount of time expected for 100% of susceptible mosquitoes to die. And the diagnostic dose, these two terms are very closely intertwined. And so this is the dose of insecticide that kills 100% of susceptible mosquitoes within the diagnostic time. And like I mentioned earlier, it's going to be the concentration of insecticide within each bottle in the bioassay. And so certainly these two reference points, diagnostic time and diagnostic dose, are really important because they're the points that all field mosquito populations are compared to. So you may be wondering, you know, what am I going to use for my diagnostic times and my diagnostic dose? In the CONUS manual, the CDC actually provides diagnostic doses and diagnostic times for susceptible laboratory colonies for various mosquito species against many technical grade insecticides. So I certainly suggest if you don't really have the ability to rear your own susceptible mosquitoes to be able to get your diagnostic time for your bottle bioassays, I certainly suggest using the diagnostic times provided here for susceptible mosquito populations. As I mentioned, my lab just uses the diagnostic doses provided by the CDC here. So we use all of these diagnostic doses in the bottle bioassays that we conduct in my lab. So if you can use the suggestions from the CDC to determine what diagnostic times to use. You can also rear your own susceptible mosquitoes to get your diagnostic times to use. Another thing that is a good idea is, so let's say that you're just starting out conducting resistance testing for a population. It's a great idea, you know, just even if you don't have anything to compare that population to the results of the resistance testing for that population, just go ahead and get those results, because that's then going to be your baseline data. And so you can then compare the results you obtain from future resistance testing to that baseline level of resistance if you're not able to compare your resistance testing results to anything else. So that is always a place to start, is at least getting that baseline data so you can compare future results to that data. So I keep talking about mosquitoes, obtaining mosquitoes to use for your bioassays. And so you may wonder, well, how am I going to get those mosquitoes if that's not something that you're doing on a regular basis? And so this slide gives you some ideas of the types of, mosquitoes that can be used. So certainly the bottle bioassay can be performed on mosquitoes collected from the field, or those reared in an insectary from larval or egg collections. So, sorry. So I'm going to start actually here at the bottom. And if this is all you have, if you're only able to collect adult mosquitoes to perform your bottle bioassays, that's fine. That's really okay. You want to try and collect at least, try and get about 125 mosquitoes per bottle bioassay that you're going to be performing. And that's going to be about 25 mosquitoes per bottle. And you have 4 treatment bottles and 1 control bottles. One thing to be aware of, though, if you are just using field adults, that you're not going to know their ages. They're going to be different ages. They're also going to be in different physiological classes. So some might be blood fed, some might be unfed, some might be gravid. So you're just going to not know that information. And so it may introduce some variation into the results that you see for your bottle bioassay. But certainly what you want to do is, even if it's after you conduct your bottle bioassay and you've killed all of your mosquitoes to be able to count them, you're going to want to identify all the species that were used within that bottle bioassay. And really it's best to select, if there was a predominant species present in that, in the adults that you did use for the bottle bioassay, it's best to try and use that predominant species to get the results about. So if there's 95% of the mosquitoes that you've tested belong to one species, then just consider using that as the species that you've tested, and the results for the bottle bioassay can be considered adequate for that predominant species. So, so certainly getting adults, you can use field caught adults for this. Additionally, you can, if you have the ability to rear mosquito larvae to adults, you can get larvae through dipping. And just the benefit of rearing your mosquitoes from larvae is that you're going to know the age of each of the mosquitoes within the resistance testing. And it could be easier for you to go ahead and identify the mosquitoes before conducting the bottle bioassay. So potentially you are able to use all of one species for that bottle bioassay. But if you don't have the ability of rearing mosquitoes, then certainly, you just can't do it. And then, of course, this is taking it a step further. If you are able to do this, you know, it's great to rear eggs from ova traps. Additionally, it's, you know, about the same level of reducing variation in the results you might see in the bottle bioassay, because you are going to know the age and the physiological state of the adult mosquitoes that you are using for the bottle bioassay, because you've reared them from egg, and so you know how old they are. And again, using eggs or mosquitoes reared from eggs can help you, can make it less work later on for you identifying the species and or separating out the species in your bottle bioassay. And the reason that I talked about it being good if you can determine the age of your mosquitoes that you're going to be using for the bottle bioassays is because you can see in this graph that here are, let's see, there's going to be the susceptible mosquito population is represented by the green line, and then the red line and the yellow line are the same field mosquito population, but here you can see what the mortality rate was for that field population at 3 days. And then you can see what the mortality rate for the field population was at 15 days. So mosquito age, and so it looks more like a susceptible population. So mosquito age does have an effect on esterase level in metabolic resistance, but you don't necessarily know if the population you're testing has metabolic resistance. Even though it would be nice to know the age of your mosquitoes, it's all about in what capabilities you have. If this is not feasible for you, then it's not feasible for you, and that's okay. So if it is feasible, you know, it's great to use mosquitoes that are approximately 3 to 10 days old, and you're certainly welcome to use both males and female mosquitoes, adult mosquitoes, when in the bottle bioassays. So just another thing to consider when you are preparing to conduct bottle bioassays, think about what insecticide or insecticide formulation you're going to use for the bottle bioassay. So certainly if you're getting the CDC bottle bioassay kit, you can request all of the insecticides from the CDC, and those insecticides that you're going to get are going to be technical grade. But should you use insecticides other than the ones provided by the CDC, I recommend using technical grade insecticide products in your bottle bioassay. And the reason for that is because it's a goal of the bottle bioassay to determine insecticide resistance to an active ingredient. And here we're going to use permethrin as an example. But in many of your formulated insecticide products, they're going to contain additives. And the point of those additives is to increase the efficacy of the active ingredient. In this example, we're talking about permethrin. So if you're using a formulated product in your bottle bioassay, you may not actually be seeing the resistance to your active ingredient because you have different additives that are also present in that formulated product. So you're not actually potentially seeing the resistance specifically to permethrin. And that is really the goal of the bottle bioassay, is to determine the mortality to that active ingredient and not towards the formulated product and containing the additional additives. So just another thing to keep in mind, I mentioned this earlier, you know, field testing is certainly very beneficial after you conduct and determine or detect resistance in your field population using the bottle bioassays. Just know that the bottle bioassays, they're not meant to correlate with operational control parameters. So there's the insecticide dose within the bottle bioassay, the diagnostic dose, it's not going to equal a label rate. So only the case mosquito field trials are going to be able to tell you what you're going to see in terms of in the field against formulated products. Okay, so we've touched on what the bottle bioassay is, what resistance is, where you can get all your free stuff. So now it's time for to cover the actual demonstration of the bottle bioassay. And rather than me just talking about it and you seeing pictures on the PowerPoint slides, my lab members were kind enough to participate in a demonstration of the bottle bioassay steps. And so that is what I'm going to show now. And this demonstration is based on the CONUS manual for evaluating insecticide resistance in mosquitoes using the CDC bottle bioassay kit. And if there are issues with the video sound or quality, please let me know via chat, and I'll do my absolute best to fix this. Before I show this, just remember that the people in this video, we all work at a medical entomology lab, and we, you know, we're not, this is not Hollywood, so there's going to be some issues with the filming. You may see some inconsistencies between the different shots. So there's going to be, it's imperfect. So I hope you actually enjoy the imperfections and you still are able to gain benefit from watching this video. And this was filmed during the pandemic, so you'll see everybody in the video wearing masks. Let me know if you have issues with volume. Bioassay procedure. You need to make the insecticide solution you'll be using to coat your bottles if you do not already have it made. To make the insecticide solution, use the clear 50 ml conical tube provided in the CDC bottle bioassay kit and measure out 50 milliliters of acetone or absolute ethanol. This acetone or ethanol, they do not need to be pure or research grade. They can be purchased at a drug or grocery store. My lab uses acetone. Whoa. Okay, so I apologize. We seem to have some buffering issues. Cut. Before you begin the CDC bottle bioassay procedure, you need to make the insecticide. Acetone or ethanol, they do not need to be pure or research grade. They can be purchased at a drug or grocery store. My lab uses acetone in our bottle bioassays, so from here on out, I'll just refer to the acetone in the procedures. Just know that absolute ethanol can be used instead. So you're going to use one of the disposable pipettes provided in the CDC kit and transfer 1 milliliter of acetone to the amber vial containing technical grade insecticide. Mix several times by pipetting up and down and transfer the mixture from the vial into the conical tube. You're going to do this about 3 times to make sure that that is mixed up. Now transfer the acetone left in the clear conical tube to the amber colored conical tube. Now you want to make sure that you use the pipette to get out any remaining insecticide solution from the small vial and put that into the amber colored conical vial. Next, it's time to put the lid on the conical tube and put the parafilm around the lid to make sure that it's nicely sealed. And you will mix up the insecticide solution by inverting it about 5 to 8 times. And of course, it's important to label your conical vial or tube containing your insecticide solution with the name of the insecticide. And once that solution is made, it can be stored in the refrigerator for future use. The next thing you need to do to prepare for performing the bioassay is labeling your bottles with masking tape or colored lab tape and a marker. I highly recommend using different colored tape for your control bottle versus your treated bottles. And if you're performing multiple bioassays at the same time, I also recommend using different colored tape for each insecticide. So once you've got your tape ready, you will use your marker to label the bottles and also make sure that you label the caps of each of your bottles as well. And this step is really important because you don't want to accidentally put a treated cap on your control bottle. So on these treatment bottles, you're just going to label them with your insecticide name and the numbers of 1, 2, 3, and 4. And again, you want to make sure that you're writing the corresponding number for the bottle, also on the cap of that bottle. Then to make sure you don't accidentally use the pipette with insecticide residue to add acetone to your control bottle, it's a good idea to go ahead and label a disposable pipette provided in the kit
with the name of your insecticide, and then also label another one with control. Once you've got your bottles labeled, you can go ahead and coat them. Starting with your control bottle, remove the cap from your bottle, and using the pipette label control, add 1 milliliter of acetone to your control bottle. Then, using your pipette labeled with your insecticide name, malathion for this example, add 1 milliliter of your insecticide to each of your bottles. Also, after you've added the insecticide solution to the bottle, make sure you screw the cap on tightly to that bottle. Even though it's not shown, you're going to coat the control bottle the same way that I'm treating the insecticide solution bottles here. And what I'm doing here is I'm twirling the bottle around to make sure that that insecticide solution is totally covering that bottom. And then I'm inverting the bottle and allowing the insecticide solution to pool at the shoulder of the bottle. And then I spin it around to make sure that the entire shoulder area is covered with insecticide solution. Next, I'll completely invert the bottle and allow that insecticide solution to pool inside the cap of the bottle. And I'll make sure, by spinning it, you're making sure that the cap of the bottle is completely coated with the insecticide solution. Now it's time to make sure you are coating the sides of the bottle well. So lay all the bottles on their sides and gently roll the bottle so that the sides all the way around are coated. Next, you're going to remove the caps off your bottles and continue rolling them until all the liquid has evaporated from the inside. My lab likes to roll the bottles with their caps off for a solid 3 minutes, just to make sure that everything is completely evaporated from the bottles. But you don't have to go that long. Just make sure that all the liquid is evaporated from the bottles. So now if you're going to be performing your bioassay the same day, you want to let your bottles rest for a little bit of time, usually about an hour, to allow those fumes to come out of the bottles before adding mosquitoes. We generally don't run assays the same day. We like to coat our bottles in the afternoon and then put them inside of a cabinet and let them sit in the dark overnight, and then come back the next day and use the bottles for the bioassays. So whether it's an hour later and you're going to perform your bioassay or it's the next day, the first thing you need to do is aspirate mosquitoes into all of your bottles. I like to start with the control bottle and add the mosquitoes to the control bottle first. And then add approximately 25 mosquitoes to all of your treatment bottles. And of course, this goes for the control bottle as well. You don't have to be exact with that number, It's an estimate, and you can add both male and female mosquitoes. It's helpful once you add the mosquitoes into the bottle just to lightly put the cap on, and you can come back later and screw it on tightly. This does have to be done pretty fast so that you don't have mosquitoes flying out, but you want to make sure that you don't blow the mosquitoes too hard into the bottle, because that can actually kill them. It's certainly possible to release mosquitoes during this process, and generally we have mosquitoes flying around in my lab. Once you have the cap screwed on your last bottle, go ahead and set a timer either for 5 minutes, which is when you're going to need to take your first mortality reading, or set a timer at 0 and just let it start running. Also look at the clock and write down what time you started. Now you want to take a look at each of your bottles and check to see if any mortality was caused during the process of transferring the mosquitoes into your bottles. And this reading will go at time 0. This is your time 0 mortality reading. Also on your data sheet, you want to record the environmental conditions in the lab, and that can help explain issues that you may see later in your bottle bioassays. Now you are 5 minutes into your bioassay. If you set your timer for 5 minutes, go ahead and reset it for another 5. Or if you just started your timer at 0 when you began your bioassay, make sure that you do take this 5-minute mortality reading. And to do this, you're going to count the number of dead or alive mosquitoes in each bottle, whichever is easiest. Generally, it's easier to count the number of dead mosquitoes when you're first starting out with your readings. So those readings that are done at 5, 10, 15 minutes. Now you are 10 minutes into your bioassay, and you need to again record the number of dead or alive mosquitoes, whichever is easiest, in each bottle. So if you set your bottle or your timer for 5 minutes, go ahead and reset it again for another 5 minutes. It can be helpful to use a permanent marker to mark a tick mark at the bottom of each bottle. And at that tick mark is where you can start counting your dead or alive mosquitoes. Rotate the bottle, counting them. Once you get back around to the tick mark, you know that you have counted all your mosquitoes that are alive or dead, whichever you're trying to count at the moment. A dead mosquito is one that cannot stand, has erratic behavior. It might fly up and then all of a sudden fall down to the bottom of the bottle. And this is indicating that the insecticide has reached its target site. So when you're recording a dead mosquito, the mosquito does not actually have to be stone cold dead. So if you tap the bottle and mosquitoes can't right themselves, or they're having difficulty flying, then that's going to be counted as a dead mosquito. Now you are 15 minutes into your bioassay. If you're resetting your timer after each mortality reading, reset it for another 15 minutes. go ahead and take your 15-minute mortality reading. So again, you're going to record the number of mosquitoes that are alive or dead, whichever is easiest. And then this is actually going to be repeated every 15 minutes until all the mosquitoes in your treated bottles are dead, or up until 2 hours. Yay, your bottle bioassay is finished. So either you're at 2 hours or all the mosquitoes are dead in your test bottles. If you have live mosquitoes remaining at the end of your bioassay, and you certainly should in your control bottle, a great way to kill them is leaving the caps on the bottle and just putting them in the freezer for at least 10 minutes. It may even be good to leave them in there for about an hour, because you certainly don't want the mosquitoes flying back up into your face as you're trying to count them. So once all your mosquitoes are dead, you want to dump them out into separate petri dishes and start the process of counting the mosquitoes that are in each bottle. It's nice to separate out males from females and have a count for the number of males and females in each bottle. Add the number of males and females together in a bottle to get your bottle total. Once you have the total for each bottle, you can then go back and fill in the dead or alive numbers at each time period using that information. Now take your data sheet and go to the computer using the Excel spreadsheet provided by the CDC and enter in the mortality at the different time points, as well as all other information for your bioassay. As you enter your data, a graph will automatically be created of your mortality over time using a linear scale. Your bioassay is complete and you have a beautiful graph of your mortality over time. Now all that's left is to clean up. You want to wash your bottles with warm soapy water and then rinse them thoroughly at least 3 times with water. And you can just use tap water for the rinsing. In my lab, we use Alkanox, the detergent, to clean the bottles. And we usually let the bottles sit filled with hot soapy water overnight. And then we come back the next day and rinse them 3 times with the tap water. You want to make sure that your bottles are completely dry before you try and coat them again. If you want to ensure that your cleaning process is adequate, introduce some susceptible mosquitoes into a recently washed and dried bottle. The mosquitoes should not die right away. If they do, repeat your washing and drying procedure.
Whoop. So I know no one wants to watch that again, but I hope that you're able to gain some information on the different steps within the process. And I hope you enjoyed the video. Certainly it was not the best video made ever, but if you want to nominate us for Emmys, you know, that's fine too. We worked really hard. So I also now just want to talk about some additional information for you to consider, and also some tips for you to, I just want to give you some tips for performing the bottle bioassay. So the first thing to think about is correcting for control mortality. And during the video, I explained that when you're adding the mosquitoes to your bottles, you can certainly get mortality. And so while, you know, at the end of your bioassay, if the mortality percentage is greater than 0 in your control bottle, that's okay. In most cases, mortality up to 3% in the control bottle can just be ignored. But if there's a mortality between 3 to 10% in the control bottle, you want to use Abbott's formula to correct the findings. And then if the mortality in the control bottle is greater than 10% at the end of the bioassay, then the results of that particular run should be discarded, and then just go ahead and repeat the bioassay. But let's say that the mosquitoes you collected for that one bioassay that has control mortality greater than 10%. They're just, you can't get them again. Then it's, you know, you can also just use Abbott's formula to correct for the control mortality when it is above 10%. But it is recommended to just go ahead and repeat the bioassay if that rate is greater than 10%. This is provided in the CONUS manual, and then also in the other manuals provided by the CDC, so you can look that up separately to refer to that. So this, I just wanted to touch more upon this, and it was mentioned in the video. So after you coat your bottles, you want to let them dry with the caps off. If you're not going to be using them soon after coating them, you want to, when you're storing them, make sure you store them in a dark place. So if you're going to be using bottles that day and you're just letting them rest for like an hour, you can just place like a towel or maybe a cardboard box or something on top of the bottles to keep them dark. And really that's because these insecticides are, they can be photosensitive, so you want to keep them protected from light so that the light does not break down the insecticides within the bottle. If you're not going to be using the bottles the same day, if you're going to be using them the next day, you can just store the bottles in a drawer or a cabinet, and they can be stored from 12 hours to 4 days depending on the insecticide. For Nalid and pyrethrum, they do break down quickly in oxygen, so they should be used immediately after they're prepared. For organophosphate coated bottles, you should use those within 2 to 4 days. For pyrethroid coated bottles, use them within 1 to 3 days. And if you've let a bottle sit and you're not sure if the insecticide solution is still going to kill mosquitoes, just go ahead and introduce some susceptible mosquitoes into that bottle. If they die in the expected time frame, so at the diagnostic dose, then you know that that bottle can be used against your field population that you're wanting to test. When you're making up bottles for your assays, they can be used for more than one batch of mosquitoes in the same day. What's going to limit your ability to reuse the bottles, it's going to be humidity being built up inside those bottles. As you introduce mosquitoes, you think about blowing into the bottles, blowing the mosquitoes into the bottles, you're introducing humidity into the bottles. So just make sure that if you are reusing the bottles, wait at least an hour to 2 hours between running the assays before introducing more mosquitoes. If you're going to be reusing the bottles the following day, just go ahead and again leave them with their caps off overnight in a dark place. So in South Florida, humidity is a really big issue. So we generally do not try to reuse the bottles in my lab. So someone asked about this in the chat, and I do want to touch on this, giving some information on what to do if you do find resistance in your population. So you can try and determine the mechanisms responsible for causing the resistance. And some of these tests are going to be pretty complicated. For example, molecular testing, enzyme assays. And so I'm not going to discuss the complex methods, and I'm just going to quickly discuss the KDR phenotypic assay and the inhibitor bottle bioassay, because I feel like mechanism testing could be a whole webinar unto itself. So I'm not going to take a whole lot of time on this issue, but I certainly do want to touch upon KDR and inhibitors, because when you're performing your bottle bioassays, conducting these tests to investigate your mechanisms, they don't require a whole lot of extra time and work. And so it would provide you to get the ability to gain some data on mechanisms without you having to do too much additional work. Whoop. So I accidentally have a slide repeated. Moving on. So first, to investigate if KDR is responsible for the resistance that you see in your population, and this is of course going to be only against pyrethroid insecticides. You can conduct the KDR phenotypic test, and what you're going to do is, this is really easy. You're going to run your bottle bioassay like normal and record all of your time mortality points, and then at the end of the 2-hour bioassay, instead of freezing or killing your mosquitoes in order to be able to count the numbers, what you want to do is transfer them into clean holding cups and let them hang out in the clean holding cups for 24 hours. So use one clean holding cup per bottle, and we usually give the mosquitoes sugar solution soaked cotton ball on top of the container to provide them with some type of sugar to feed on during this time period. So at the end of the 24-hour holding period, all you're going to do is count the number of mosquitoes alive, and then once you do kill all your mosquitoes, you're going to be able to come up with the percent of mosquitoes that were alive, of course, at your diagnostic time, very important. But then specifically to investigate KDR, you're also going to have your mortality at 2 hours, and then get your record your mortality at 24 hours. And then all you want to do is compare mortality at 24 hours to 2 hours. If your mortality at 24 hours is lower than your mortality at 2 hours, then you know KDR is being expressed in your population. And that is a very simple way to investigate KDR in your mosquito population. Also, one thing you can do is look at phenotypic metabolic resistance, and you will run your bioassay normally against your insecticide. So I'll use permethrin here as an example, and going to run your bottle bioassay like normal, and you'll get that time mortality curve. And if you find resistance in your population and you want to determine if that resistance is due to metabolic resistance, then you'll go beyond that one bottle bioassay with your active ingredient for that population, and you will coat one bottle there's 3 different enzyme inhibitors, and each of the enzyme inhibitors correspond to a different enzyme class, metabolic enzyme class. So you want to coat, if you want to investigate whether or not all 3 enzyme classes are contributing to metabolic resistance, go ahead and coat one bottle for each of the different metabolic or for each of the inhibitors, and I have those listed on the next slide. So you'll have one bottle if you're trying to investigate if there's oxidase activity contributing to your resistance. You'll treat one bottle for that inhibitor. If you're trying to investigate esterase activity, you'll treat another bottle for that inhibitor, and so on. So you're gonna let the mosquitoes hang out in those bottles for an hour, and then you'll let them recover for another hour. And then you're gonna run your bottle bioassay like normal. So this is an example here of the steps you're gonna take. And then what you're gonna do is compare that original mortality curve that you saw with your insecticides. I use permethrin as my example, to the mortality curve that you see once you perform, or once you expose your mosquitoes to that inhibitor, and then run that bottle bioassay again. And this chart just gives you an example of the possible outcomes you could see. So in line A, the exposure to the inhibitor basically abolished the resistance to the insecticide. So now the mosquito population is looking more like a susceptible population than a resistant population. For line B, there's really, the resistance is only partially abolished, and here, actually no, sorry, I got that backwards. Here, this is going to be that there's really no effect of the insecticide, or sorry, of the inhibitor. And then here, this will be that the inhibitor is partially affecting the ability of metabolic resistance to occur. And again, I just want to thank Janet McAllister for allowing me to borrow her slides, and I accidentally forgot to remove the CDC little emblem there, so it just gives me a chance to give a shout out to Janet. So here is what, after you perform your bottle bioassays with your inhibitors and you're comparing your mortality curves between when you expose your mosquitoes to your insecticide, and then versus exposing them to each of the different inhibitors and then conducting the bottle bioassay with your insecticide. Here is what you can, what it can look like. This is an example of an Aedes aegypti population that is from Miami-Dade County. And so in the black dotted line is going to be the time mortality curve for the susceptible Orlando Aedes aegypti population. So you can see that this population is susceptible to permethrin. They died very quickly when exposed to the permethrin. So next, if you'll see, this black line represents the field population of Aedes aegypti when no inhibitor was run with it. So this is just the regular permethrin bottle bioassay here. And you can see the time mortality curve. There is certainly resistance being observed here. And so each of the different lines here represent the mortality curves that were achieved by exposing that population one at a time to the different inhibitor assays, the different inhibitors. PBO is going to inhibit oxidase activity. DEF is going to inhibit esterase activity. And then DM is going to inhibit GST activity. So you can see that when the activity of these three different enzyme classes were inhibited, then the mosquito population did become closer to looking more like that susceptible Aedes aegypti population against permethrin. So certainly in this example, I think you can say that the three enzyme classes are all contributing to metabolic resistance. And one thing I will say is, if you are wanting to investigate metabolic resistance and you don't feel like you have enough time to run all of these assays, go ahead and at least run the assay with PBO, because a lot of formulated products with permethrin and also sumethrin will contain PBO. So so at least if you run that one, you will, well, first, if you find that resistance is in your population and it's due to metabolic resistance, then you're going to be able to know if the resistance is largely due to oxidase activity or not, and if the PBO and the product you're considering using is going to be able to overcome that specific resistance mechanism in your population. Okay, so I'm going to end this webinar with just a quick example of how I've used the results from bottle bioassays to make adulticide recommendations. I'm going to provide you with this example. This is actually something that's currently taking place. So for those of you who do not know, currently we, in the state of Florida, we're experiencing a West Nile virus outbreak. Sorry, trying to put all those words together. So as of a week ago, there were a total of 48 human infections that had been reported for this year. I'm sure there's actually more infections now because this data is a week old. So 17 of those cases were symptomatic, and certainly the symptoms of West Nile virus can be very serious, and they can, have neurological consequences. So, and this is something that's very important that's happening right now. So 48 of those 48 exposures occurred in Miami-Dade County. So as part of the resistance testing project for populations in the state of Florida that my lab is currently conducting, we are running bottle bioassays with Culex quinquefasciatus, southern house mosquito, that were collected in 8 different areas of Miami-Dade County, and running them in bottle bioassays against 6 different active ingredients. So here you can see what the results look like for one of the populations, the pinecrest population. And so just looking at this chart, you can see that the population is resistant to permethrin and etifinprox and deltamethrin. Also, you know, that cutoff, it's 89% mortality observed for malathion. So I guess you would, you know, that's considered being resistant to malathion as well. And then comparing the mortality at 2 hours to 24 hours, you can see that there is knockdown resistance exhibited in this population towards etifinprox here, and then for permethrin here, because your percent mortality is lower at 24 hours compared to the 2 hours. So when making adulticide selections or making adulticide recommendations for Miami-Dade to use during this current West Nile virus outbreak, I saw that chlorpyrifos, the mortality for chlorpyrifos at the diagnostic time was 93%. So in my opinion, this could be the most effective adulticide to use against this population. Of course, there's so many things to consider when choosing an adulticide for a population. Certainly when we're talking about an organophosphate, it's going to be, is your community acceptable of you using organophosphates? Certainly, you know, I can't say that, I just make suggestions, but this chlorpyrifos was the insecticide that did result in the highest mortality rate at diagnostic time. Next, we had malathion that had a mortality rate of 89% at the diagnostic time. So this could potentially be another option. And sorry, let me just minimize this chat for a second. And then though we saw mortality to the 4 different pyrethroids that were tested, when you looked at the mortality rates at the end of the 2-hour bottle bioassay, there was differences in the mortality rate at the end of the 2-hour bottle bioassay for the 4 pyrethroids. There was a mortality rate of 71% for permethrin. So out of those, I would say that permethrin is going to be the best pyrethroid choice. And it was certainly because we did see resistance to permethrin, and there are permethrin PBO products available. I would recommend to follow up the permethrin bottle bioassay with the inhibitor bottle bioassays to determine if the permethrin PBO products could overcome the oxidase activity. And then because there is a mortality rate of 71% rather than 100%, which is really what we'd love to see, you want to try and use the management strategy of saturation to hopefully kill out the mosquitoes that are not resistant. And so you want to use the highest app rate possible. Of course, per label instructions, label is the law. And then for the other 2 pyrethroids tested, I would say that the management strategy could be used here, is just reducing the use if possible. So moderation, and hopefully this will allow keeping those active ingredients available to the mosquito control program for the long term. And again, you know, these are just the bottle bioassays, and it's certainly recommended to conduct field trials to determine the actual field efficacy rates that you're going to see with your products in the field. And for long term, you know, I certainly recommend rotating pyrethroids with organophosphates because we really only have, at least in the US, we only have so many chemical classes available to us too. And when you use the same insecticide class over and over and over again, you do have the potential for resistance to increase. So I know that, again, it's, you know, there's a lot of things that go into making a decision about which adulticide you're going to use, but just for resistance management purposes, recommend rotating the pyrethroids with organophosphates could potentially reduce resistance rates from increasing in populations. And as I mentioned at the very beginning of this, the bottle bioassay is just providing the mortality of a mosquito population at a certain time. And so it's a snapshot. So while this is the results that we saw now, and these are the recommendations I have based on those results, you know, it's recommended to perform this testing at least once a year because, you know, mosquitoes aren't static and resistance rates aren't static. So I know that we are at the end of our time. So I've provided the link to the exit survey. I will pull up the chat and go ahead and share that in the chat as well. So hopefully you guys will be able to access it. Really, it's, while I would love for everyone to answer it's the exit chat is certainly just needed by the people that are trying to get or attending this webinar to obtain the FDACS CEUs for the public health pest control license. So certainly if you do fall into that category, please make sure that you take the exit survey. Sorry, got to pull up the chat to do that. Here we go. And honestly, I just have one more slide left that's thanking people. This webinar was scheduled to run from 1 to 3, and it's certainly very close to 3 o'clock now, but I have time for questions. If anyone has questions, this does not have to end right now. I'm very happy to take questions. I expected it to run till about 3 o'clock and then need to take additional time for questions. So please feel free to go ahead and put questions that you have in the chat. And I'm now sending the survey. And so I'm going to now start looking at some of the questions that were asked. I'm pretty sure Janet McAllister from the CDC is currently present in the webinar. So Janet, oh, look, Janet, okay. Janet said to make sure to remember to rinse the top and bottom of ampules when mixing solutions in the CDC kit. So thank you so much, Janet, for contributing, and please contribute anything else. So no, I do not feel that using bottle bioassays that's the correct method for comparing formulated products. That's going to be using a field trial. That's going to be the appropriate way for evaluating which is going to be the best formulated product to use. And that's because, as I discussed earlier, the bottle bioassay is meant to detect the best performing active ingredient. In formulated products, you don't only have an active ingredient, you have lots of potential additives. So that's why I don't feel like the bottle bioassay is the best method for that. So why are male and female mosquitoes used in bioassays when only females transmit pathogens? Excellent question. It's because both male and females are contributing to the genetics, and genetics are what leads to resistance. So, and of course, environmental factors. So that is why you want to use both male and females in your bottle bioassay. Whew. So, how does the CDC bottle bioassay compare to the WHO assays? Personally, I was taught how to perform the CDC bottle bioassay. So I cannot answer that question for you. Janet McAllister, if you want, you know, I don't really think that there's no reason to try and compare results. You know, you pick the assay that for you is going to be the best to use, and use that assay to detect resistance in your population. But if Janet wants to contribute to that question, I certainly would love her to. And if anyone, okay, so Sarah asked, do you recommend using F1 mosquitoes for testing? Oh, okay, Sarah. So I think you want to use whatever mosquitoes whatever population of mosquitoes you're gonna have enough of for testing. We try and collect enough field mosquitoes to conduct the testing. Generally, we do not have enough eggs for that. So when we hatch them to complete the testing, so yes, we will blood feed and then get the F1, sorry, I need to unmute Janet McAllister so she can talk. Thank you, Janet. May take me a minute because there's so we generally use either parent generation, so the generation straight from the field up into the F2 generation, and let's see. Huh. Not being successful at unmuting you, Jan. I'm so sorry. Okay. All right.
Okay.
Here we go.
Can you hear me? Perfect. Sorry, I didn't want to interrupt you on that the answer you were giving me, but the previous question about the WHO test and the CDC bottle bioassay. The CDC bottle bioassay is kind of unique in that it's a direct measure of a toxicological parameter. It's the time it takes that insecticide to get to the target site. The WHO test is calibrated based on dose mortalities. And that's a little bit different measure. It's an indirect measure of resistance, and that it's they do use a diagnostic dose, and they do use a endpoint time period in their test, but all of the calculation behind the dose and the time is based on different dose mortality curves. And so they measure slightly different things. And then, but the WHO test, if you have a long history with it and have used it, certainly it's a good test. They just measure things slightly differently. And with the WHO test, you don't get that additional information about the strength of the mechanism because you're putting your mosquitoes on a paper that you have to purchase, and then for a certain amount of time, and then you hold them for a certain amount of time. And a lot of people tend to get a lot of mortality in the test as they're running it because you are holding your mosquitoes often for 24 hours. So that's the real difference between the two.
Excellent. Thank you so much, Janet, for answering that question. So if you have any other questions about this criminal or conducting resistance tests, please ask now in the chat, and either I or Janet will be happy to answer your questions. Okay, I'm not seeing any more questions, so apparently this was very informative. So excellent to hear. This is again being recorded, and you will be able to access this later on FMEL's website. I want to thank you all for attending and putting up with all the technology issues, and thank you for your patience. So thank you so much.
Okay.